Hello, hi. Um, Let me see. Okay, I think this works. Good morning, good afternoon. I am Kavita. I'm from the Williams Lab, and I just want to give an overview about some of the current ideas we have in the Williams Lab about molecules and memory. And I think memory is something very inherent in the origin of life, but it's very implicit. We haven't quite defined it, and hopefully, in the next few minutes, I will try to demonstrate how we're trying to do that. So before we go to the origin of life, I want to just break down the word memory. When we say the word memory, we think of all these different ideas and concepts. We think of remembering people, dates, names. Biologists often associate neurons with memories, some of these molecules. Memory is often a word associated with computers as well, where we have short-term, less short-term, and much more long-term memories. At the heart of all of these processes, though, is this idea of acquisition, encoding, and retrieval of information. Information of different kinds, but the process itself is the same across all these different formats. Another definition of memory, though, is the ability to record information about events and the facility of recalling them at a later point. Now, this definition is really interesting for biology because biology is amazing at remembering things. So biology is amazing at recording information, and biology is amazing at recalling them. And the most, I guess, amazing part of biology is genetic memory or nucleic acid memory. We have information about genes stored over millions and millions of years. We have information stored as identity of these specific molecules here, ACGT, U in case of RNA. And this form of storage and this type of information stored in nucleic acids is termed as heredity, and it's an excellent form of memory in biology for millions and millions of years. What I want to demonstrate today is that maybe it doesn't stop there. Maybe it goes beyond. And when I say beyond, we mean beyond genetic memory. We talk about non-genetic memory. We see that there are ways in biology which are programmed to record information which doesn't quite reach the genetic level. And this is often as histone modification, DNA methylation, RNA changes, structural as well as molecular changes in the RNA, and a host of other chromatin modifications as well. And these are forms of memory which are less long-term, but there are still ways in which biology records information for a period of time. Now we're going to push this a little further and present a couple of other instances in biology where memory is recorded, but we don't traditionally associate with memory. And we want to suggest that maybe these are some of the ways in which biology records. Cell signaling pathways, so MAP kinase pathways, are ways in which cells regulate homeostasis mechanisms, where cells are capable of detecting their environment, detecting their situation, and regulating upregulating or downregulating downstream pathways. Allosteric regulation, which is a much more short-term form of memory, where we have small molecules regulating specific metabolic pathways, and feedback loops, of course. So these may be called short-term memory, but these are also, we posit, forms of memory in biology. Pushing that even further and going even smaller into the molecular scale, we want to present things like prion-based inheritance. Now, prions are basically proteins, misfolded proteins, which are independent of sequence. So they essentially have very less to do with the particular sequence of amino acids, sequence of genes that code for them. They are essentially structural pieces of information which can last up to sometimes even longer than which can last generations, sometimes even longer than epigenetics or DNA methylation. So there are this is prion-based inheritance. The second one is quorum sensing, where we have a different set of live bacteria which are capable of detecting populations and regulating their behavior accordingly. Now, this is regulation, but we would actually argue that this is a form of short-term memory which can last across generations and provide the said organism with some kind of an advantage or disadvantage against the environment. And I think this is where we want to push this boundary even more and say, okay, if biology has so many different forms of memory which we don't traditionally think of, maybe memory is something much more inherent to biology, and maybe that should influence our study of the origin of life. And hence we are working currently within the framework that the origin of life is the origin and evolution of sophisticated molecular memory, which goes way beyond heredity. This is not something completely new, and I would actually say that all the research into the origin of life, beginning from the RNA world, which was, I think, in the 1970s or 80s, has been trying to look for some kind of memory, some kind of system, self-replicating system that records memory, that can transmit it to the next generation. We are currently looking at changes in the identity of the molecules, where we have backbone changes, we have identity of the bases being changed. And these are essentially, I think, attempts by the origin of life group to try and identify what kind of memory existed long ago. And although they are all amazing pieces of research, I think we also need to open up our mind to the fact that memory was maybe something way beyond heredity. Maybe it was less tied to the identity of the molecules, something more to do with the environmental processes, the regulation, the recording of information in different ways. The Williams lab right now is working on a system where we can study the evolution of memory, which is non-hereditary, non-genetic memory. And we are studying this within the framework of chemical evolution, although we're studying this within the framework of pre-Darwinism, pre-Darwinism, which is non-genetic memory. I'm not going to go into too much details right now, but I just wanted to give an outline of some of the ideas in lab today, and they are essentially when you cycle a group of small molecules in a wet-dry cycling phenomena, they show the capability of recording environmental events, like this one here. So when we cycle these group of molecules in two different conditions, this specific environmental event was recorded in the HPLC spectra of this memory. Now, this is independent of the individual molecules, independent of the identity of these molecules. This recording is not dependent on the specific structures, but this is an emergent phenomena. And I think this is something really interesting to know, and I think this should inform future research into the origin of life. With this, I would like to finish my presentation. Thank you.
Great, thank you. Do we have any questions from the audience or online?
Thank you, Kavita. That was a great talk. Could you go back one slide? Okay, so
yes.
Could you just explain a bit more about how you're recording the environmental events?
Absolutely, yeah. So the chemical system we are exploring right now is essentially a chemical evolution of these eight different components, where we wet-dry cycle these 8 components and study the formation of wet-dry cycle these 8 components at 45 degrees and study the formation of polymers through different analytical techniques, one of which is HPLC, which is essentially this graph writer is an absorbance at 210 nanometers of the different cycles of the different aliquots taken at different cycles. Now, when the same system was run in 2 different conditions, one of which had a perturbation, slight perturbation at one specific cycle and everything else was the same, we recorded that when we compared the spectra, we had a significant perturbation in the HPLC. And this perturbation faded over time. So when we ran this for maybe 15 or 20 cycles, the spectra, then there's no difference between the control and the perturbed system. So this is something really, really interesting, and I think this is actually a path-dependent behavior and demonstrates memory.
Very cool. And we do have a question from Blue Jeans, which is, do we also understand how new memory is attached to nucleotides when there is evolution in organisms? Are there any specific chemicals involved?
I am not too sure about that. I would imagine that I guess creativity in the structure of nucleic acids today would be very difficult. But maybe without the framework of life, there may have been many more chemical groups attached, functional groups attached, and much more creativity, that's for sure.
Agreed. Thank you. Any more questions from the room?
I'm also curious about this last little experiment that you are talking about, where you see persistent effects from perturbations that stick around.
Mm-hmm.
So I'm just looking at the graphs there, and am I interpreting what the peaks mean? That when there are peaks somewhere, it means that there is some kind of a substance that has come together from the smaller pieces? And if so, is it sort of like not much is happening, not much is happening, then suddenly a bunch happens, and then it sticks around from that point forward?
In the simplest terms, yes.
Okay.
Since this is a different spectra,
Right.
I have essentially...
different spectra. Okay, I missed that.
So this is a differentiation of, so I've subtracted one from the other, but if this wasn't a different spectra, yes, an individual peak would mean a bunch of molecules.
Okay, I missed that part.
That's fine.
Yeah. So okay, so the okay, that makes sense. Cool.
All right.
I'll go here.
Can you go back to your, I think you had an origin of, or a life definition or something up there?
Yeah.
Okay, so it's like life is sophisticated molecular memory, right?
Yes.
From that. So you gave the example of like prions as a good
Yes.
example of like what you kind of like are thinking about. So are prions alive? I'm mostly just kidding with that one. That's but yeah, it's a cool idea.
Absolutely, I think prions are really interesting in their structure and in recording events independent of their sequence. I think that was something that is pretty amazing.
This is a great talk. Thank you. I was just curious, how long do you run the experiments for? Like, how much time is being captured in the spectra that you show? Yeah, the cycles. Yeah.
At this stage, we only have up to cycle 15 for this specific experiment, but we definitely plan on doing longer cycles set up in lab right now.
But how much time? Oh, I see. Sorry, I just couldn't see from my... Yeah, how much time per cycle?
Oh, sorry. It's 2 days per cycle at 45 degrees.
Okay. Okay, thank you. And I'm assuming you're interested in expanding the experiments to longer and
Absolutely.
longer
We have a grad student who's working in our lab who's planning on extending this phenomena much further, as well as looking at other emergent properties of the system.
Thank you.
Thank you. So we have to move on to the next section, but thank you.